Om Shri Sai Ram. The second speaker of today's session is Dr. Mrs. Rachna Sachwani. I am proud to introduce my own student, Dr. Mrs. Rachna Sachwani, who is currently working as a senior scientist at Life Science Lead, Millipore Sigma, Sheboygan Falls, Wisconsin, United States of America. Even before introducing her as a senior scientist, I wish to say that she was an outstanding student of Sri Sathya Sai Institute of Higher Learning. She received gold medal from the divine hands of our beloved Swami in the year 2001 for her outstanding performance in BSc Honours Chemistry. Dr. Rachna, a technical expert with a unique background in synthetic and analytical chemistry, received her PhD degree from the Indian Institute of Chemical Technology, Osmania University, Hyderabad. She had received senior and junior research fellowships in the years 2005 to 2007 and 2003 to 2005 from CSIR. Mrs. Rachna received Dr. Reddy's Spirits of Excellence Fellowship Award by Dr. Reddy's Research Foundation, that is DRF, Hyderabad in 2002. She worked as postdoctoral research associate from 2008 to 2010 at the University of Illinois, Chicago, USA. There she has synthesized and evaluated a variety of transition metal catalysts for vinylation of oximes. After postdoctoral research, she joined as organic analytical chemist in Cediburg Pharmaceuticals, United States of America from 2011 to 2014. In 2014, she joined as a scientist in custom process development, Millipore Sigma, and continued till 2019 as scientist. Currently, she is a senior scientist in Life Science Lead, Millipore Sigma. She has a good number of publications and presentations. Dr. Rachna is pursuing Black Belt certification and has completed 6 Sigma Green Belt certification. As your teacher, I am very proud of you, Rachna, for your achievements. Now, I welcome you, Dr. Rachna, to the session. Sairam.
Sairam, my humble pranams at Divine Lotus Feet, and a very loving Sairam to everyone out there who is listening. There was a fabulous talk by the previous speaker, and I feel like that's a perfect segue into what I am going to talk to you about today. And before I do that, let me thank the Pavna ma'am for a kind introduction. And I did have a slide here, but other than being a scientist and currently the OPEX manager for my organization, I am a proud mom of an 8-year-old, and I have a wonderful husband. And like many of you, I started my journey of chemistry at Anantapur campus, where Rajni ma'am and Pavna ma'am and many other teachers taught me. And that's where my love for chemistry started. So I owe it to our institute and of course to our, the divine hand behind us, no matter where we go. So when I was thinking about all of you, when I got this opportunity to be coming and talking to you, I was thinking of what should I be discussing. And I have to say that there were 3 things in my mind about what I want to talk to you about, right? One is there something which I did not know as a student and I would have liked to know about, right? The second thing was, what is the need for the future industry? As you guys, I understand many of you who are listening to me are either doing your bachelor's or master's or maybe even your research. What would be something which pretty much each one of you can use in some way And then third is, of course, what do I bring to the table if I discuss a topic like that? So with those three things in mind, I chose this topic to talk about. And the title of my talk is Process Analytical Technology as a Pathway to Lean Chemical Manufacturing. Now, if you look at this topic, uh, there are two keywords here, right? There's process analytical technology and there's lean manufacturing. So I'm going to go over those things in detail. I have tried to keep my talk more simple. I don't have too much data to share, but what I'm trying to bring home is that if you can understand these two concepts, you can pretty much apply them at any walk of your life, in future. So, so to go about it, I will first discuss lean, which is a very, very general concept. We will understand what lean is and why should lean be used. And while doing that, I will also discuss the 8 forms of waste, which are important for manufacturing. And then we will talk about more chemistry, right? And that is the process analytical technology, where we will compare the traditional ways of analysis compared to the PAT approach, which is a more recent approach, very apt with the title you have for the symposium, the recent advances in chemical sciences. We will discuss the approaches PAT uses for the analysis. And then we will learn some tools and sampling systems and some details of how PAT should be implemented in manufacturing or in research. And at the end, I will discuss all the benefits for PAT and lean, and hopefully would get some interaction from you in forms of questions or other ways. I second the sentiment of the previous speaker that we are missing that aspect of interaction because we are doing this virtually. But I do hope to stay connected with you, and you can always reach out to me, if not today, then later. So, so let's start with lean. So lean is a way of thinking. That's how I look at lean, right? So when the simple definition of lean is that you eliminate all the waste and make your life better. Now, some of you might be thinking, what do I mean by waste? Hold that thought, because I am going to discuss that in a little more detail in future slides. But before I go there, I want to tell all of you that lean is for everyone. No matter what you're doing, it's an important concept to learn. So many of you are chemists today, and I hope you continue to be chemists and bring a lot more research, like some of the talks we have heard during the symposium, on the table. But even if you choose to go in a different field, like go to finance or marketing, or even decide to just stay home and raise wonderful kids, you can still use the concept of lean. And why do I say that? There are two reasons for that. The first reason is that each one of us participates in processes every day of our lives. So when we go and work, right, we are always part of a process. So let me give you an example, right? Every morning I wake up and I have this goal of making to work on time, let's say by 8 o'clock, right? So I have a process to make sure I reach my work by 8 o'clock in the morning, right? So there are certain things I need to finish before I can get there. And that and that is a process. Same for each one of you. Whatever you do, you have certain processes you follow during the day, and that's how you get things accomplished. So if you follow a process, you can use the concept of lean. The second reason why lean is for everyone is because we all engage with customers. Now when I say this, some of you might be thinking that what do I mean by that, right? So in our mind, when we think of customers, we think of maybe like a customer service where like you are at a call center answering some questions to somebody who needs help. But the real definition of customer is whenever you deliver something to someone, that person is your customer. So in my example of me getting up in the morning and making sure I reach work on time, I am my customer. Okay. So I am my own customer. For you all, as you're taking some classes virtually, and I was just talking to Rajni ma'am this morning, and I found out that you all have been very good at attending your virtual classes. So when you do attend your classes, you your teacher is your customer because you are delivering your focus to your teacher. At the same time, for your teacher, for your professor, you are the customer because they are delivering the lesson and they are imparting the knowledge to you. So all of us engage with customers every single day, and that's why lean is important for us to learn. So the definition of lean is it's reduction of waste and all the non-value added activities based on customers' perspective. So whenever we are thinking of lean, we are thinking of, okay, everything we do, every activity we do as a part of a process should deliver value to the customer. If it's not delivering value to the customer, it is a non-value added activity and should be eliminated. So, for example, right, I go back to my example of waking up in the morning and trying to get to work on time. Now, I do need to have, let's say, good hygiene. So, I need to get my shower. I need to have a good stomach full so that I can be focused at work. So, that's a value added activity. And I need to drive to work. So, driving is a value added activity. Now, if I spend 15 minutes looking at my phone or looking at some forwarded messages on WhatsApp, that is a non-value added activity, and that needs to be eliminated to have a lean process in the morning. uh, so I'm talking about value and I'm talking about waste, right? And the goal of lean is that the value should go up and the waste should go down, right? So you should reduce and minimize the waste. And the value is always defined based on customer perspective. So when I talk about chemical manufacturing, and believe it or not, but right now this is a very, very critical topic for all of us because, as you all know, the world just went through a major pandemic, and vaccine is very much needed. And vaccine was made in a record time, and the way it was done is by eliminating all the non-value added activities from the process, and that's how they were able to deliver a vaccine in record time, right? So we need to make sure that the quality still stays up because customers want quality, the speed is good, and the cost is appropriate, and those are all contributing to the value. But then we have some waste. And how do we define waste? So I come back to the topic as promised, and there are 8 different forms of waste based on the lean philosophy, and they're remembered by the term Tim Woods. So T stands for transportation. uh, Whenever you're moving items or information, it's considered a waste. If you have a lot of inventory of products which is not delivered to the customer, it's considered a form of waste. If you are having access movement in your workspace, that is also considered a form of waste because it's not adding value to the customer. If your product is waiting, or some information is being waited upon, that is again considered a form of waste. If you over process your product, so I go back to the chemical manufacturing. So suppose you made a compound, right? And you need to do extractions, and you need to do one water wash to get rid of your water soluble impurities. But instead, you're doing 3 water washes. So those 2 extra water washes are over processing, and that's considered a form of waste. Uh, Making more than what you need is also considered a form of waste, which is overproduction. So the reason we consider that as a waste is because instead of making more of something we don't need, we could be using those resources to make something we need. So it's considered a form of waste. And then defects, any mistakes we make in making a product in pharma, that's also considered a form of waste because either the product is rejected, which cannot be used for the desired purpose, or it needs to be reworked, which means extra resources needs to be spent to purify that product. So that's why defects are also considered form of waste. And last but not the least, uh, not using the workers to their fullest abilities is also considered a form of waste. So skills is the last form of waste. So these are the 8 forms of waste which we try to eliminate whenever we try to make a process lean. And as I mentioned before, lean is a concept which is a general concept, but now, uh, but nevertheless, it improves efficiency by eliminating waste. But now we are going to discuss lean in uh, in PAT. But before that, I can't help but discuss and remember our college routine. So if you talk about you know my student time, it's been almost 20 years that I left Anandpur campus, but I still can say with proud that every single activity we had on the college was a value added activity, right? And starting from the morning prayers and exercise to the self reliance sports, every single thing we did during the day added value. And that's a great example for lean philosophy. So now I come to the actual topic of process analytical technology. And the way I would like to approach this is that first I want to share with you what traditionally happens, right? So if consider any pharma company, and I'm saying pharma because that's the most common form of chemical. There are many chemical manufacturing which happen, which are made, but the most common form of manufacturing is pharma, which everybody is aware about because it goes into pharmaceuticals. So the way it happens is there are certain operations which are mentioned here on the slide, but as those operations are happening, so you get the raw material, and then as you're making the product, as you go through different steps of synthesis, extraction, distillation, at every step, the product needs to be checked for the quality. And the way it is checked is we sample it and then send it to QC lab, and then QC lab, quality control lab, tests it. And then if the test passes, it meets the specification, then the product is used for the next step, or if it is the final product, it is packaged and shipped out. But if it does not pass the specification, then it is rejected, in which case it's either reworked or discarded based on the situation of the product, right? So now if you look at this traditional approach of pharma manufacturing, uh, I just spoke to you about 8 forms of waste. So let's see how many different ways we have, right? We have transportation, we have motion involved here, where we are taking the samples to the QC lab. We are using skills, like a skilled operator is taking the sample, so we are underutilizing our operator. At the same time, while the material is getting tested in QC lab, uh, the product is waiting, it's either getting over processed or the inventory is getting built, as we are making more because we know that this is going to take time, so we make more to cover for that gap, right? At the same time, if the QC lab rejects it, then it results into defects, so because the reaction has already happened, so really you can't change much about it, so then it's a defect. So there are all 8 forms of waste which are present in the traditional analysis approach of pharma manufacturing. So, what does PAT do, right? PAT, which is process analytical technology, suggests that we test the material right there. So, as we are going through all these different steps, we have the analytical instrument attached right there, and we get real time data, uh, and that way, we are able to take care of all of those 8 forms of waste. And that's how PAT is considered a very, very lean approach to pharma manufacturing. This is a fancy definition of PAT, and I'm not going to read it. But one point I do want to bring across is that other than removing the 8 forms of waste, as you are doing process analytical technology, you are building quality in the process, because you're watching your process the whole time. So you are uh, building quality in each lot through timely measurements instead of measuring it after the fact. So that is the beauty of PAT, or process analytical technology. And again, to explain, the conventional product oriented analysis happens at a snapshot. So if you think about a process, a compound, which uses three steps to make it, right? So after every step, you would sample it and you will test it, right? So you're basically taking snapshots of the process and seeing what the material looks like. While in PAT, it's recommended that you are testing it the whole time as the process is happening, as shown by these arrows. So you're testing it the whole time. And how do we test it? There are three different approaches to the measurement. We can do it at line, which means you you isolate the sample from the process and then analyze it in the process environment. The other option is to do it online, where you divert the sample from the process and analyze and return it back to the line if it is appropriate. Or the third way, which is the most efficient way, is the inline analysis, where you are analyzing the sample in the ongoing process. So this could be more like a probe or a sensor you can have in your reaction vessel or in the reaction line, where you're constantly testing the sample. So there are these three different approaches by which the analytical approach is made. So the one thing where PAT really, really stands out is that as we do the traditional analysis, for us, the process is actually a black box, because we take samples at certain points and test them. So we know that, okay, after the first step, this is what the product looked like. But what was happening during that step is not known, right? So that's why process is a black box for us. Whereas if you use PAT, the process is understood and mastered, because you have the complete data of what the process looked like at every single step. And that's why PAT is highly encouraged as a regulatory approach. So now, uh, what comes to your mind when that if you are trying to do inline analysis versus the offline analysis, what's the most critical thing? The one most critical thing is the choice of sampling points, right? Where do you sample? And here I have some examples. Whenever any chemical compounds are made, these are some of the common equipment which get used when you're making the process. So you have a reactor in which you do the reaction, right? Then you might have a place where you blend the chemicals, like a powder, blender. You have a place where you dry the chemicals, and then if you're formulating it into tablets, then there is another equipment we use. So these are some of the basic equipment. There are many more fancy equipments out there, but some of the basic equipments. And one thing to remember is that it's very, very critical to choose a proper sampling interface. Okay, and here in a nutshell, I have given some examples of what are those sampling interfaces which can be used. So you do one, of course, the most preferred one is an insertion probe, which you can either put directly in the reactor sampling system, or sometimes it's even inserted in the drain valve. Okay, so that's the inline analysis, where you're directly inserting the probe in the reactor vessel. The other option is a fast loop. So you have a loop coming out of your equipment, and that's where you insert your analytical tool. Now, this is a good way to do it, but sometimes you have reactions which are running at high pressure or high temperature, and then it's not physically possible for us to have an analytical probe in the fast loop, in which case we divert the sample to a slower loop. and this loop will be at a more like room temperature, normal pressure, and then you're able to insert your probe or analytical tool right here to the analysis. So one critical thing to remember is that failure of the sampling interface is responsible for most of the problems in process analytical applications. So if you all ever take a route where you're trying to use PAT for any of your research, or any of developing any process, make sure that you spend enough time looking at sampling interface and trying to use a proper sampling interface, and then you will have successful data collection. So on this slide, I'm trying to talk about some common PAT tools. And as students, I'm sure you are learning all different analytical techniques. As you go forward, you learn many more. So I'm not going to really discuss any of these techniques in detail, but I did want to drive the point home that as you have different steps in the process, the needs of analysis might change. So, for example, when you're doing a reaction, your focus might be on completion of the reaction, what the impurities look like, what is the kinetics of the reaction, and what is the solvent composition. So, all of those analysis, there are some typical techniques which are mentioned here, are very, very useful to understand the reaction completion and impurity profile. Now, as you're doing the workup of a reaction, you're working up a reaction, then the yield becomes more important. The impurity still stays important. In that case, you could use techniques like chromatography, MIR, NIR. Now, once you have worked up your reaction and you're trying to crystallize it, right? You're trying to isolate your solid in form of crystals. At that point, the particle distribution shape or form or supersaturation, those become more important. You try to watch those. And at that point, turbidity and FBRM, PVM, Raman, MIR, those are the common techniques. And on, right? So for dryer, you have different techniques. And then for the final milling, when you're trying to grind your particles, again, FBRM seems to be the technique of choice. So there are all these different techniques. And here I have mentioned some of the more often used techniques in PAT. So there are techniques like mid infrared, which are very, very versatile and very easy to interpret. So it seems to be the most popular technique used in PAT. We have Raman, which is limited in sensitivity, but still seems to be a popular tool, especially because it can work with solids and slurries. We have NIR, which is easy to deploy, but it does need some expertise. So it's a little bit more tricky to interpret it. So you need some experience with the technique to be able to use it successfully. And then UV. UV is, as we all know, all the small molecules, especially the drug molecules, typically have a chromophore. So in that case, UV can be a great tool. So you can try to use UV analysis, inline analysis, and understand your process better. So now, when I say that, right, when I talk about all these techniques, some of you might be thinking that, oh, so that's all PAT is about. Instead of using an analytical equipment in a separate lab, I'm using it right there. Is that what it is all about? Not really. There is more to it. So one thing we are not considering here is that when you analyze a sample in the lab, you are actually cleaning up the sample, right? So you're preparing the sample, you're cleaning it up and then analyzing it. And you have a lot of knowledge about the sample. Whereas when you do PAT, there are so many variables in place. Plus, you're not cleaning the sample, you're analyzing it as it is. So the way PAT approaches it is, if you think about all the variables which a reaction is facing, you might have a reactor configuration which could be different every time, right? You could have raw material qualities which are different, and at the same time, you based on the time of the reaction, in the beginning of reaction, you could have a high concentration of starting material. Middle of the reaction, you might have maybe high concentration of one of the intermediates. And towards the end of reaction, you might have a higher concentration of product. In that case, when all these variables are happening, we use chemometric techniques to understand and extract the information in a relevant way. So to explain it a little bit better, you are using not only the analytical chemistry knowledge when you do PAT, but you're also using statistical modeling. And one of the common analysis technique used is multivariate analysis, where the data is reduced and by the dimensionality, and then we are able to draw correlations and patterns. So that's how PAT becomes a successful technique. And there are very advanced softwares actually available which could help us with some of this modeling. So here I have one simple example. So if you look at this data here, right? This is a Raman PAT data for a very, very simple reaction where chloroformate reacts with alcohol to give carbonate. And the PAT tool and the multivariate analysis was able to draw the patterns and correlation and give out this data where we are able to see the difference as if you look at the test 0 and test 3, these samples were also tested by the traditional techniques, and you look at the 0, you are slowly seeing your starter disappearing and your product appearing, and of course during the solvent wash you're seeing an high axis of the solvent. So this is one great example how PAT can give you a whole picture of what's happening in the reaction. Not only that, as you use PAT, it is also giving you a lot of mechanistic insight about what could be the mechanism of this reaction, and that's where PAT again becomes a very, very knowledgeable tool. Uh, so I hope with all this explanation, I'm able to drive the point home that when you do PAT, you not only collect your data, but you save the data at every single point of the process. Of course, you run many batches. You have data from many batches, and the multivariate analysis actually can relate to the best batch data and the current batch data, and is able to give us a full perspective of how robust our process is. At the same time, if something goes wrong in the process, we can have a feedback control where we are able to correct or the process as it is happening instead of finding it out after the fact. So PAT has many different aspects to it. It helps us understand the relevant factors in the reaction. It helps us with the process analysis. It uh, it uses the multivariate analysis to get a full picture of the process and also the mechanistic insight. And at the same time, it can help us for a better process control where we are able to control our reaction from going in a direction, an undesirable direction, where we may not want it to go. So, how do we implement PAT? There are two paths for implementing PAT. Either you can choose to implement it right from the R&D stage. So whenever we are doing development on a molecule, we are deciding how to make a molecule, we can use PAT as a tool, or we can use it directly in the manufacturing. And there are definitely uh, different opportunities whether we use it in development or manufacturing. Some of the important things to think about is the overall purpose of PAT, right? So if we do it in development, we should do it more for understanding the process. Whereas if we do it in manufacturing, we should do it to analyze the trend and also control your process. If you're using it in development, it's highly recommended that you use multi-component analyzers because you're not familiar with the process, right? So you might want to analyze everything. You might want to check the pH, you might want to check the pressure of the reaction, you might want to, check the UV data, IR data, and everything you can possibly check to understand that process better. But if you're doing it in manufacturing, chances are that you already understand your process, in which case you can use more targeted as analyzers. For example, if you know that your reaction does not really change in pressure, you can just have, but the pH is a critical criteria, you can have an inline pH meter, and that should be enough for your PAT. Now, one other difference between implementing it in development or manufacturing is the data complexity. So, for development, definitely the multivariate approach is important because there will be a lot of variables and you're trying to understand all of those variables. But if it is manufacturing, depending on your process, if it is a simple process, it's okay to use univariate analysis. If you think there is more challenge to the process, then definitely go for multivariate analysis. Uh, Also, when it comes to the expertise in PAT, for development, you do need more expertise in method design and development, whereas for manufacturing, just the operational and maintenance expertise is enough to operate PAT successfully. So so the drivers for your path one versus two, right? The path one is if you want to implement it in R&D phase, it enhances your process understanding and control, and it also increases your risk management. So you can develop a safer process if you use PAT, because then you have a complete understanding of the mechanism, and you can figure out what factors could affect the safety of the process in large scale. But if you are doing it in directly in manufacturing, then you should have the goal of maybe improving the process based on the data collected, and also to eliminate any variability in the process, because now you have all this data, so you can make sure that your process stays consistent from batch to batch. So that should be your driver to implement it in manufacturing. And one of the places where PAT definitely brings the most value is in continuous manufacturing. So, in this picture, I'm trying to explain the batch process versus continuous process. And for those of you who may not be aware, I will uh, just explain the common difference in one way, is that in batch process you do things at a large scale where you're mixing, say, like the reactant A with B in a big vessel, and the whole reactions happens at a time. Whereas in a continuous process, you are mixing reagents slowly, so you're doing kind of like mini reactions in pipes. And that's where, if you use PAT, uh, and if you realize that something is not working or something is not going well in your reaction, you can stop it right there, and you're not losing any inventory, right? Because you stopped it right when the problem happened. Now here in the batch process, you could still have potentially a big kettle of mixture of reactants where something hasn't worked, and you still have to deal with it. So that's why PAT brings a lot more value in continuous manufacturing, and it also has a very smaller footprint. So that makes it a very attractive option for continuous manufacturing. Uh, so with all this information, I hope I'm able to drive the benefits home. So PAT helps us with faster analysis time, and it is definitely important for a faster result time. As I told you, we are cutting off a lot of extra things which we don't need to do if we have inline analysis. It also helps with faster decision making, and especially with the current pandemic situation, having faster decision making and faster results is obviously very, very welcome. And last but not the least, it also helps with a greener analysis. So because you're using less time, less resources, maybe less solvents to do the analysis, so it's a greener way to do the analysis. Some of the other additional benefits of PAT are sustainability. So as I mentioned, because you are reducing the energy requirement and you're doing real time in process analysis, so you're saving a lot of time on analysis. You're using less chemicals to extract, to filter, to wash the product. So you are definitely using less chemicals there, and at the same time you're making a process safer, because now that you don't have to sample process, process at every step, you are saving a lot of time and making your process very safe. So you are not exposing your operators to those chemicals. So that's another big advantage of PAT. Uh, while doing making it a safer and sustainable process, we are also putting nice controls in place, right? So you are ensuring that the batch to batch consistency is maintained, which means that you are opting for a better quality of product. And it also helps with the stability, because if you take the samples, go get them tested in QC lab, uh, the molecules are very labile, and your stability of the material could be compromised sometimes because of the weighting in the analysis. So you're also taking advantage of the material stability. And last but not the least, it's a big favorite of FDA or any other regulatory agencies, because it makes us understand the process better, and it enables continuous verification of the process. So that's why regulatory agencies really like the use of PAT. So with all this in mind, I think I do want to conclude uh, with a very, very important survey here. So we have data from Agilent Technologies, which actually says that of all the time which spent is QC lab, when the sample goes to QC lab, only 6% of time is spent on the analysis. There's 61% of time which is spent in sample processing. There's another 27% of the time which is spent in data management, and 6% of the time in collection and distribution of samples. So if we use PAT, we are basically getting rid of all of those steps, sample collection, transport, preparation, and thus PAT is a no brainer for a lean thinker. So I do want to again stress the fact that no matter what you do, you should be able to use these two concepts. Think of, as research scholars, try to think of how you can develop a lean process. Every step you do has to be value added. At the same time, see if you can do analysis on the go instead of trying to do it after the fact. And that holds true in any part of our lives. As we continue in our journey, we constantly analyze every situation. So that's how PAT is again relevant there. And to conclude, a lean thinker does not think why the glass is half full or why the glass is half empty. It always thinks why this glass is twice as big as it should be. So with that, I do want to share some of the references here. If you want to know, there's a lot of data out there. I couldn't share much of the data because of the proprietary issues, but there is data out there which you can look into for PAT and, of course, lean philosophy. And at the same time, I do want to thank all of you for patiently listening to these topics. I hope you'll be able to make use of it. I want to also give my special thanks to the chemistry department at our university. And I want to thank my organization, where I have Merck, where I have learned all the concepts of lean and PAT. And I do want to thank my family, because as I was preparing for this presentation, they tried to give me a lot of time and accommodate during the holidays. And last but not the least, I thank Swami, who is my ultimate teacher and guide at every moment. Thank you.
Thank you, Dr. Rachna, for that lucid presentation on such a general topic like lean and PAT, yet so important for all of us. Here are a few quick questions for you from the students. The first question is, can you very quickly give us an example from your personal industry experience from your personal life?
Uh, about PAT?
Yeah.
Okay. So, so one again, I can't give like the molecular details, but one great example is about a product which is very very complicated to make, and we have been making it for many years in the batch process, and it solidified on us all the time, and we couldn't really figure out. And we would test it, at different stages where we'll take a sample and we would try to understand it, and it did not really give us like consistent quality. So for many years we have made the product and we ran into problems again and again. But not too long ago, we ventured into continuous manufacturing and PAT, and now we are able to make the product, as I said, in very, very small steps, right? So we are mixing our, let's say, reagent A and B gradually, and as we are mixing it, we check it at every stage, and we are using Raman for that. So, and what we have noticed is that there were these many hot points which were causing all the issues. So now that we have been testing it throughout, the moment we see the pattern where the exotherm happens, we're able to control. So we're able to control it at the point, at very localized point, and we're able to make our product very successfully. So that's one of the examples, which again I highly recommend looking into literature. There are a lot of cool examples where PAT has been used, and especially for like Grignard reactions, for example, right? And Grignard reactions are a little bit hazardous reactions, but PAT can be used to make sure that the bromine alkyl bromide accumulation doesn't happen, which can be a safety hazard. So you kind of monitor it through PAT and make sure that you're not losing the product. Mm-hmm.
Sorry. Can you explain to us how to apply lean in a research laboratory? How do we avoid or minimize over-processing of the product in the initial stages of research? As in the initial optimization of the product, there is a lot of wastage of chemicals.
Right, and that's a very great question. And I would suggest to start with some of like really, really basic PAT tools. In university, we obviously have limited infrastructure, so some of these probes can be expensive, right? But what I would suggest is that maybe start with some basic tool, like have maybe do one reaction where you spend time and you're continuously taking the IR of the reaction, right, from the beginning to end. At the same time, you're also monitoring, of course, depending on your reaction, the temperature, pressure, and pH density. Those are very, very common parameters, but they can tell you a lot about the reaction. So maybe continuously monitor it. And then you have a great mathematics department there, yeah, right? Ask them to help you with some mathematical modeling where you can there are some softwares, right, like where you can relate all that data, and then come up with so if you do it, my suggestion is if you do it maybe 2 or 3 times where you have enough data, you will be able to draw patterns, and then you will be able to make sure that for example, right, if you are processing your experiment, you're trying to get rid of an impurity. If you're constantly collecting the data, you would know where this impurity is going. Is it converting into something else as you're working it up, or is it changing? Is it just going in your water wash? So if you're doing a constant data collection, you will be able to get some of those hints, and then you will be able to design your process based on that, instead of just assuming that okay, 2 water washes and now I test my final sample, it does not have impurity, that means I needed 2 water washes. So yeah, so that's my suggestion. I also recommend actually using some of the vendors. The vendors are really good at letting you use probe. That's how we did it actually. They let you use probe for like, let's say 6 months, like for testing purposes. So you can use their probe for free and learn from there. So that's another way you could potentially use it. And in the bargain, you become a PAT expert. So there's a high demand for PAT experts in the industry, just so you know, and that's why I chose this topic.
Ma'am, you've heard a lot of pros about PAT. Uh, are
Mhm.
there any cons?
Hi, good question. I think I kind of said that. So one and the one actually there are two major. So one con is understanding the proper sampling portal, right? And I mentioned that if you do not choose a good way to use your like your sampling portal, that will affect your PAT data. So that's one. And then the other thing is that in large scale manufacturing, right? So in research, I wouldn't say there's no con because you are collecting, you are understanding your process better. But when you go to manufacturing, the other con is that you are trying to use your process in a very big, especially if it is a batch manufacturing, it's a big vessel, there are a lot of other equipment around it, so there are a lot of vibrations to it, and some of these probes can be very sensitive to vibrations. So I think that's one con which we are still trying to overcome in some of our processes. But that doesn't mean it's not possible. It just means that you need to have a proper setup to protect these probes from vibration so that your data doesn't get affected and you get like reliable data. So those are some of the cons. But again, my solution to that is to go to more basic PAT, right? Like, as I said, density, turbidity, pressure, temperature, those are very basic things. They will not be affected by these vibrations and they still tell you a lot about the process.
One last question, ma'am. Uh, One last question, ma'am.
Sure.
How PAT can be used to decrease the usage of toxic chemical substances in the manufacturing of medicines and other essential daily life healthcare products?
Good. Yeah, that's another great question. And the way I look at it is that if you are doing PAT, right, you are checking your sample as you're making it. So if there is a toxic impurity which is being made, you know right away. So suppose in research, right, you have 5 different ways you can make a compound. So you start by this one way and you see that if you're using PAT, you will see that, okay, there is this particular peak I'm seeing which forms, and this is a toxic compound. You can right away decide to change a process and use maybe another route. Instead of what happens is that we develop these processes and we already work it up, we check our final product, it looks good. But then it's much later we find out that these compounds have some toxic impurities, and so they cannot be successfully used, for medical purposes, or there's a lot of improvement needed before they can actually be used. So that's another way, you're looking at it. And that's again, the multivariate analysis is a great tool because what it does is it takes all the data you have collected. For example, like if you're collecting the ramen data, you would have data at every single wavelength. It takes all of the data and literally uses it in different dimensions and does a modeling and tries to tell you all the correlations and all the patterns. So some of the things which you could have missed if you just ran one single ramen on your sample, you took a sample and ran it, you could have missed it. But now with all so many data points, like if you have a 24-hour reaction, suppose, right, every 2 minutes is collecting data, so you have so many data points, you are able to compare and contrast and figure out what's forming, what's not forming, and that can give you big clues into toxicity of compounds.
Thank you, ma'am, for this beautiful elucidation on the concept of lean and PAT. I'm sure it will be
Thank
very useful to our research scholars here for planning out their experiments, and we look forward to actually many more interactions with you so that we can actually learn more from you on this particular concept. Thank you, ma'am.
Same here. And thank you so much for the opportunity. It's absolutely wonderful to come and talk to our students. And we all know that there's always a special bond between us no matter where we go. So thank you for bringing me back. Thanks,
Sarah, ma'am.
Sairam.